Hello students, myself Pritish Kulkarni, biology educator, here to explain you the very important topic from the genetics unit, and that is nothing but replication of DNA. So guys, as you know, you are aware about the thing, how much important the topic genetics is. So for the examination of NEET, genetics is one of the most important unit, and if you want to score good marks, you should remember the concept of replication for the purpose of NEET. So as you know very much, when you study any topic with me, we have learn with fun. So guys, without wasting time, today we are going to study the very important concept, and it is replication of the DNA, which is also known as DNA duplication. So guys, what is the need of it? First of all, you must be thinking, what is the need of replication of the DNA? So you know, when cell has the particular division, cell has to give the same copy of its genetic material to its progeny, and hence cell has to replicate its DNA first. So yes guys, that's why I have written over here, it occurs during the S phase of cell cycle in eukaryotes. So I want to tell you, if whatever we are going to discuss in today's session, each and every point will be used for NEET exam. And the topic what I have written over here, it is up to the concept of class 11th and 12th. Because as you know, DNA replication is there in BSc, MSc, but you must be thinking, I'm not going to go much in that detail because I know what kinds of questions have been asked in the previous 10-year examination. So yes, without wasting time, we'll start with simplifying the DNA replication process with me. So guys, first of all, as I told you, it is the doubling. It is the formation of two daughter molecules. So yes guys, first of all, there is a parent DNA molecule. So you know that DNA molecule is a DNA double helix. It has direction, you can say that, running anti-parallel to each other. So if you can just imagine the DNA double helix, it is having two strands which are running anti-parallel to each other. One is running 3' to 5' and the another one is running 5' to 3'. So you have the concept of DNA double helix in your mind. Now, what exactly the mode of DNA replication is? So in NEET examination, they will ask you this question. So you please remember, DNA replication is a semi-conservative mode. So what it means? In which one parental strand is conserved and the new daughter strand is formed. So guys, you please take a note of this. The replication in which one strand is conserved and one is newly formed, so it is called as semi-conservative mode of replication. The second one I am going to explain you during the discussion of this diagram. Semidiscontinuous mode. Why it is called as semi-discontinuous, I am going to explain you in the discussion of the diagram. So guys, I have written here the major point important for the NEET. See, there are three processes. You know, there is a concept of central dogma of molecular biology, where there is a replication, there is a transcription, and there is a translation. Yes, out of that, we are going to study only one, DNA replication. So guys, when I am talking, there are two strands in DNA. Both strands acts as a template. Yes, both strand acts as a template. So what the template strand means? Template means on the strand on which there is the synthesis of new DNA strand. So guys, please take a note of this here. 3' to 5' also acts as a template, and 5' to 3' also acts as a template. This is the peculiarity of the process. So guys, please take a note of this. Both strands acts as a template. Now, the third very important part. DNA replication takes place only in 5 prime to 3 prime direction. I want it to be very important part for you people. Don't forget it. All the processes, DNA replication, transcription, translation, their direction is from 5 prime to 3 prime direction. Never forget this. So the question comes in neat. Which of the following is the correct direction? Yes, the DNA replication takes place in 5 prime to 3 prime direction. Last but not least, yes, it should be the origin part actually. So you know guys, all these processes, they have 3 steps: initiation, elongation and termination. So similarly, this process also has 3 steps. The first one is initiation, second one is elongation, third one is termination. So yes, the first step, replication starts from the sequence which is known as origin, ori. Guys, this is the particular sequence present on the double-stranded DNA where it has certain nucleotides which have easily breaking down property. Means you can say they are having reach in 80 reach sequences. A double bond T. See, actually it should get separated easily. So guys, now I am going to explain you how exactly this replication starts. So as I already explained you, there is a first step called as initiation. So during this, the origin is the main sequence. So guys, you know that there is replication in prokaryotes, eukaryotes. There is a slight difference. So as I told you already, the prokaryotic DNA replication, eukaryotic DNA replication, there are certain differences in the enzymes used and all. But yes, we are studying the DNA replication in general. So guys, this is the first step where origin. So when the particular DNA is going to initiate the replication, the particular strands of the DNA will get unwound. So guys, the first step for the replication to begin is the enzyme oriented process. You can say enzyme catalyzed process. Yes, that's why I have written the replication enzymes and proteins. So you can look at this now. The first enzyme, yes, the first enzyme involved in the replication of DNA, it is helicase. Helicase, the one which removes the helix of the DNA. As I already told you, DNA is double helix. So if DNA has to replicate, its double helical structure should get unwound. Unwound means it should get separated from each other. So as I shown you here, the DNA was previously it was having the double helical structure and now it has unwound. So the first enzyme is helicase, which has the property of unwinding. So the coiled structure will become now uncoiled. Yes, now you know that DNA has the property of immediately binding to each other. You can say that complementarity. So A binding with T, G binding with C and all. So this complementarity will immediately have the rollover here. It may be possible that after unwinding, DNA strand again rewound. So to prevent that, there is a very important protein present which is called as SSB protein, single stranded binding protein, which will go here immediately and starts binding these single stranded DNA. You can see like this. So they will form a chain of proteins, SSB proteins. And yes guys, they help to prevent, you can say, the binding of two strands again, or you can say that SSBs are responsible for stabilization. Because in NEET examination, they will ask you the question of functions of each and every enzyme. That's why. So helicase, the function is unwinding. SSB function is to stabilize DNA. Now, you know that when there is an unwinding continuously going on, there is development of certain strain over here. You know, in this particular part, they develop a particular strain. So to remove that, yes, the third enzyme comes into picture and it is called as topoisomerase. So students, this topoisomerase gives a nick. So there are two types, topoisomerase 1 and 2. You are not having these things in detail. I am just telling you for the knowledge purpose. So topoisomerase just relieves the strain over there. So yes, the third enzyme very important. And now the main part. After this, primase. As you know that for the starting of the process, they require 3' hydroxyl group to begin the process. So that's why the first very important process which is carried out by, you can say it's RNA polymerase, which will synthesize short oligonucleotide. And oligonucleotide means 5 to 10 nucleotide base pairs. So they will synthesize the primer at the start, which will give free 3' hydroxyl group just to have the elongation of the nucleotides. So guys, please take a note of this. This is one of the most important enzyme which is required in DNA replication process, primase. It is also known as RNA polymerase, which will synthesize short primer at the start. Now, after this, the next enzyme, yes, you can say it is the most important enzyme of the replication pathway, machinery, DNA polymerase. Yes, guys, you know there is the second step called as elongation. So in the elongation step, this enzyme helps in the polymerization. Polymerization means what? You know, in the cell, there are nucleotides which are already activated. DATP, DGTP, DCTP, DTTP. So they will be taken in the process. Or that particular enzyme will bring about the addition of the nucleotides with respect to what they have on the template strand. Suppose on template strand they are having A, there will be the addition of T. Suppose on template strand there is C, they will add G. So likewise, as you know the complementarity principle, the addition of nucleotide will take place. So DNA polymerase will add nucleotides and it will have the lengthening, polymerization. Yes guys, now I want to explain you the concept of this. Why it is called as semi-discontinuous mode. So have a look at this. I have written here the two names of the strand, leading strand and lagging strand. So what are they? Guys, as I already told you, DNA replication runs in 5' to 3' direction. So you can see here there is a continuous synthesis in this direction on the strand which is 3' to 5' acting as a template. So the strand which is synthesized continuously is known as leading strand, and the strand which is synthesized in a discontinuous fashion. So you know, dear students, this particular are the fragments which are known as Okazaki fragments in honor of the scientist. Okazaki fragments. So guys, please take a note of this, and as I already told you, these Okazaki fragments are 1000 to 2000 in prokaryotes, while they are 100 to 200 in eukaryotes. Yes, the question can be asked in NEET. Okazaki fragments. How many Okazaki fragments are found in prokaryotes? 1000 to 2000, and in eukaryotes 100 to 200. So please take a note of this, Okazaki fragments. So that's why the process is known as semi-discontinuous, because on one strand there is a continuous synthesis, on the another strand there is a discontinuous synthesis. So is the point clear? This is all about the process of DNA replication, which is carried out by the several set of enzymes. And the last but not least, DNA ligase. You know, it is called as gum of the molecular biology. it is going to join. So yes, once the full process gets finished, means when they come across the terminal point, as I told you, initiation, elongation and termination. So at that particular point, when the total DNA is now getting complete, and that much is to be, replicated. So there they stop the process, termination of the process. After that, this particular primates, these primers are removed, and later on these Okazaki fragments are joined to each other. Yes, but before joining, dear students, the gap because of the primers is filled. The gap because of the primer is filled with the help of DNA polymerase, and later on these gaps will be filled up by the ligase. So ligase will join Okazaki fragments and they will make a continuous strand. So is my point clear? This is the most important step in the whole process, which will complete the full replication process. Now dear students, here I have written DNA polymerase types. So as I told you, polymerase is the major enzyme in the particular topic. So here I have written prokaryotes which have which DNA polymerase and eukaryotes which have which DNA polymerase. So guys, polymerase 1, 2 and 3. In case of prokaryotes, these three are the major enzymes, out of which DNA polymerase 3 plays major role in all these activities, while DNA polymerase 1 and 2, they are considered as the repair enzymes. So please take a note of this. DNA polymerase 3 plays a major role, while 1 and 2 are repair enzymes. While in case of eukaryotes, you can see there are 5 types of subunits: alpha, beta, gamma, delta and epsilon. So guys, in case of eukaryotes, there is a DNA polymerase having all 5 subunits, while in case of prokaryotes, there are 3 different types of DNA polymerase: 1, 2 and 3. So as I already told you, out of these 3, DNA polymerase 3 is the major enzyme involved in this process, while 1 and 2 are playing major role in repair. So after this, as I told you, if something is wrong in the addition of nucleotides, because see, this is a biological process, it may happen that there may be some problem. So there is a proofreading phenomena also, where these enzymes, DNA polymerase 3 itself has an activity of proofreading. Proofreading means what? That enzyme can recognize if there is certain wrong base pair addition, it can rectify that particular mistake. So that is known as proofreading activity, and that is performed by the DNA polymerase 3 in case of prokaryotes. So guys, I hope you must have understood the total topic. I'll again just recall the things depending upon the question which have been asked in NEET. So guys, the major question which I have seen is, which of the following enzymes are used in DNA replication process? So guys, I have written very simple, as you know, I make the topic very simple. Yes, because of this, you can keep the thing easily in mind. Identify the correct sequence of enzymes in DNA replication. So guys, first enzyme used is helicase, second one used is topoisomerase, then primase, then DNA polymerase, and then lastly ligase. So I have formed a very simple sentence with respect to which you can keep the things easily in mind. He means any boy, he took pen to draw line. See how simple the sentence is. He took pen to draw line. He for helicase, took for topoisomerase, pen for primase, draw for DNA polymerase, and line for ligase. So guys, this is all about the DNA replication process. I hope you must have understood the total process in a very simplified manner. So watch my video, like it, subscribe the channel, and yes, Wish you all the best for the NEET examination. Thank you.